Couple of announcements. Hopefully I'm not spending too much time plugging the lab mutant viruses from hell course. I know that you hopefully will have a chance to start signing up for classes next week for spring term. How are we thinking about spring term already? Can we think beyond the midterm next Friday? I have trouble doing so. I highly recommend this particular lab. As we have at the bottom, it's the Cure for the Common Lab, so it's a course-based undergraduate research experience. So literally, we don't know what you're going to come up with in this lab. So if you're interested in more of the details, please let me know. I know the numbers at the bottom here are actually for spring of 2016. We'll be covering just new mutants in the spring of 2018. I'll make sure that flyer ends up out as well. There will also be a flyer for the literature review course that we'll be doing on Friday afternoons for that class, and we'll be talking about various primary literature and small group discussions, and hopefully this will be useful to some people who are interested in looking at primary literature. So that's, excuse me, this announcement. The other announcement, how many of you have seen the biology genome? How many of you should see the biology genome? Lots and lots of you. It's a great read. Well, I think it's a great read. But probably most important is one sentence right at the bottom here of our letter from the chair. And it's probably really tough to see down here. But keep an eye out for an announcement of a new summer research opportunity, the Lester Newman Scholars Program. This program is made possible by a generous donation from Dr. Newman, retired biology department professor. First Newman Scholars will be named for summer of 2018. This is going to be a $5,000 fellowship for doing lab work for a couple of people in biology. Should be announced later today, all of the details on that. So keep an eye out on the biology website and also try and post something in this class. And also, if you have questions about it, feel free to email me about those things. So without further ado, let's move on to what we're all here for, I think, hopefully anyway, is molecular biology. So let's see if I can get this thing to work. It was giving me some trouble a little bit earlier, so we shall see. Okay, so today we're going to continue talking about translation. And particularly, last time we talked a little bit about the genetic code. Today, we'll continue a little bit with the genetic code, then talk about ORFs. It's not ORCs again. Again, I think you could also have the subheading for molecular biology alphabet soup because of all of these extra things. ORF just stands for Open Reading Frame. And that's how your DNA can be read into protein. How does that happen? Through the process of tRNAs. So we'll talk a little bit about tRNAs. Again, it's one of those subjects we could spend a whole term talking about. And a lot of what has to do with tRNAs also has to do with the aminoacyl-tRNA synthetases, which I'll just call just tRNA synthetases. And we have one of the world's experts on tRNA synthetases whose lab is just over in SRTC, John Perrone in the chemistry department. Then we'll talk a little bit about the ribosome. The ribosome is really all about the RNA. And again, subheading for this course could be it's all about the RNA. And then if we get to it, we'll talk a little bit about translational initiation as far as that goes. So I showed this image last time, just going over the genetic code basically to emphasize that the code is degenerate, i.e. you can have lots of codons, which is that stretch of 3 nucleotides, which can code for any three, those three can code for potentially one amino acid that has lots of different codons for it. So the idea here, we'll take leucine as the example, where we have 6 possibilities. I'll back up here. 6 possibilities for leucine in terms of number of codons which can be used, UUA, UUG, etc. What do I expect you to remember? That there are 2 that only have 1 codon, and particularly that AUG is methionine. We'll see why that's important, because it's the start codon for most translation. And this whole process here. And just a way to read this table, which is really pretty straightforward. You start with the first nucleotide, which is the A, the second nucleotide, which is a U, and I give you one guess what the third one is. Oh, come on. It's not that early in the morning. G, thank you. So, methionine, the one I expect you to remember, say, next Friday, maybe? And no, the exam is not finished written yet. So how do we go about decoding the genetic code? So that's you reading that table, but how did people figure out how to put that table together in the first place? And so basically, all this is about, what's that first word? Translation, thank you. Übersetzen. So it's literally going from the nucleotide language to the amino acid language. And that's what the genetic code is about. And in order to do that, you need the super-duper decoder ring. How many of you know what Cracker Jack is? Shows how old I am. Okay, there's still a few of you. So the decoder ring that you find in your Cracker Jack box. Well, the decoder ring is the tRNA. And how we figured out, and we, I mean the molecular biology community, figured out that, for instance, UUU was the codon for phenylalanine, is that they were able to make RNA that was just U's, fed it into a reaction that had ribosomes in it and all the amino acids, and they just looked to see what they got out. And it turned out to be a whole stretch of phenylalanines. They did the same thing with guanines. They did the same thing with alanines, et cetera. And eventually building up the whole genetic code by putting in these synthetic RNAs and then seeing what you could get out. And this really was the development of having a reaction where you had ribosomes and all the amino acids and all of those tRNAs. You could look at them, again, put in your messenger RNA and see what you got out. So very, very useful technique, and this is basically why Gobind Khorana and a few of his colleagues got the Nobel Prize for figuring out the genetic code. So here, UUU means we have phenylalanine, and so on and so forth for the rest of the genetic code. So hopefully pretty clear that, super-duper decoder ring is the tRNA, and this is just one example over here. We'll look in more details in just a second here. So yes, we've got our decoder ring. We fed it all of the appropriate RNAs. But any given RNA can actually give you 3 different proteins. Why is that? Well, that's because the code is non-overlapping, as we talked about last time. So what codon you get, what stretch of 3 nucleotides, really depends on where you start. If you start here at nucleotide 1, oops, come on. If we start at nucleotide 1, here, AUG, which of course is methionine, and progress all the way through this particular piece of RNA, you get over to here, which is your stop codon, and this would be what we call an open reading frame. It's got a start and a stop. However, if you were to start with the second nucleotide, now you have UGA, AAG, CAA, UUU, etc. This is a completely different sequence. Now here A, the K is the same, the lysine, but glutamine, phenylalanine, lysine. So very different protein sequence here. Or if you started with nucleotide 3, you have GAA, AGC, etc. Again, very different amino acids, but still exactly the same nucleotide. So it really depends on exactly where you start. And this is what we call ORFs, or open reading frames. It depends on where you start. So if you start at nucleotide 1, that's going to give you 1 set of translated amino acids, 2 and 3. So those are the 3 different reading frames that you can have, just again where you start. And an open reading frame is literally going to be something that you have a start and a stop for, because you've opened that up in terms of translation. Now, one of the quotes from the textbook here is, "Only one reading frame contains message." That other course you can sign up for starting on Monday will tell you how that's all wrong. That actually you can use multiple reading frames to get messages from, if you're talking about cool organisms like viruses. So, questions on genetic code and reading frames. What does that mean? Time to get your clickers out. Exactly. So if there are only 2 nucleotides per codon, how many different amino acids could be encoded in a non-overlapping genetic code? 2, 4, 8, 16, or 32? And again, vote early, vote often. And talk to your neighbors, because there are people here listening and marking down whether you're talking to yourselves or me. And that's by the way, the 4 regular nucleotides that we have. I should have mentioned that. Normal nucleotides, yeah. C, A, U, and G. Ten, 5. It's getting better. We're not having this huge rush of votes right at the end. I like that. So, only 2 nucleotides per codon. How many should we have? 2, 4, 8, 16, 32. People like 16. Why? 4 squared is 16. Yes, I agree with you. Yay. Still not 100% though. We're working on that. Okay, so I mentioned tRNAs before. Let's focus in on that. It's not just like, again, the little decoder ring that you find in your Cracker Jack box. It's really an amazing molecule, and a lot of people spend a lot of time working on this, as well as Dr. Perona. There's also Dr. Awada Royal in the chemistry department. He's also very involved in looking at a lot of the modifications, particularly that happen to tRNAs. So hopefully all of you know this sort of general cloverleaf structure of RNA. This is the tRNA, which will give you alanine at the appropriate codon-anticodon interaction. These are RNAs, so just like any of the nucleotides we've talked about so far, they've got a 5' end, which is a phosphate, a 3' end, which is an OH, and it's this OH where your amino acid is attached to. And so 5' to 3', highly secondarily structured, and say secondary structure is just base pairs which are forming or interactions between the individual bases. Here, this classic cloverleaf structure is because you have 3 loops and 1 just stem here. They're almost all base paired to each other, and all tRNAs, all of them that you find in the cell have this same overall structure. They're going to differ in a couple of different ways. One, not surprisingly, is down here at the anticodon. Now remember, codon, that's what we just already talked about, this genetic code. The anticodon, just like all of your nucleotide interactions, is going to base pair. But how's it going to base pair? It's going to base pair in the opposite orientation, because base pairs only happen in an anti-parallel way. So if you look at this, going from 5' to 3', here we have IGC as your anticodon. We'll get back to I's in just a second here. This will be the first position of your anticodon, second and third. But since it's anti-parallel, it's going to be this base, the third base, that pairs with the first base of your codon, etc. So this will be a G, C, and then turns out lots of different possibilities for alanine here as well. tRNAs are between 70 and about 100 base pairs in length. So again, there's some variability there. And again, a lot of that variability is going to end up being these stems here. These loops end up being pretty well conserved. The other thing is you'll notice all of these funky things, including that I down here. What's I? Inosine. Have we talked about inosine before? Actually, once really briefly in a lecture, but basically no. So these are a bunch of modified nucleosides. And we talked about modified nucleosides when we talked about the ribosomal RNA before, where we have pseudouridine formation and omethylation, which happens. Well, what I didn't mention is the pseudouridine is this psi. So actually, there are some pseudouridines that you find in fact, all tRNAs. And this is the particular loop. You also is known as the T-psi-C loop because they all have thymidines say, pseudouridines and cytosines in them. This is just a few of the modified bases. In the case of the alanine-tRNA, it's 10 modified bases, but it's usually about 10% of the nucleotides in any tRNA are modified. Not always exactly the same way, but they're all modified. And that seems to have to do with the structure and stability of the particular tRNA that you're looking at. So clearly these things have evolved over very long periods of evolution in order to get this kind of stability to them. So this is how people always draw tRNAs. This is actually not what tRNAs really look like. They actually look like what we have here on the right-hand side. This is the X-ray crystal structure of the phenylalanine tRNA that was solved in the group of Sung-Ho Kim at UC Berkeley a little bit before I got there. But a couple of things to look at here is, yes, it's not a clover leaf, but still all of these base pairing interactions. Here is the anticodon down here at the bottom. Here's the base pairing that you have in this loop down here. We've got base pairing here, base pairing here. But now instead of them being 4 separate stems, it's really just 2, which are pretty contiguous here. Another thing to notice here is that where the anticodon is, this is, again, what's going to be binding to your messenger RNA, is quite a long way from the amino acid over here, just in space. And so what that means is that this guy has to somehow be able to talk to this guy. And that process we'll see, again, is the aminoacyl-tRNA synthetases that we'll talk about in just a second here. As if the modifications that happened in the bases weren't weird enough by themselves, turns out that a lot of tRNAs are also modified by splicing. Now, not splicing like we talked about for the last 2 lectures, which is lariat formation and all RNA-based. This is protein-based. And so there are proteins which will bind to what people call the pre-tRNAs, and again, like the pre-messenger RNA or the pre-ribosomal RNA, which will then cut and religate. So these are much more like the ligases that we've talked about before in lectures when we're talking about replication with DNA. So it's a cut and religation that happens in these. And a lot of the splicing that happens in tRNAs, or I should say RNAs in general, there's yet another lab in the chemistry department here at PSU, Dr. Niles Lehman's lab, who also is very interested in this splicing process. Just like ribosomal RNAs, tRNAs are also trimmed at the ends by endonucleases that will give an appropriate end to them. Why do all of these tRNAs seem to get so heavily modified, and particularly this splicing interaction? It's probably because they've got to be pretty darn good at doing what they do, because if they don't, translation's not going to work properly. So all of these steps, the modifications, and particularly this splicing, probably has to do with quality control. So making sure that each of those individual tRNAs are modified properly so that they can be used in the translation machinery. And the last thing, this is why I use these notes, to remind you of is we've already talked about RNA polymerase II. What does that do? Transcribes what? Messenger RNAs, RNA polymerase I, ribosomal RNAs. What are we left with? Number 3, what does that transcribe? tRNAs, exactly. So also something that I consider fair game as far as the midterm is concerned. Which of these individual RNA polymerases are going to be being used for each of these things? So Pol1, ribosomal RNA, Pol2, messenger RNAs, Pol3, tRNAs. Good. So we're all happy with that. So already looked at some of these modified nucleosides. And when we looked at that alanyl-tRNA, we had the inosine present at the first position in the anticodon. So what does that inosine actually do? What does it give us? Well, this goes back to thinking about the genetic code. You remember some amino acids have 6 codons, which are just coding for one amino acid. Well, what does that mean? Are there 6 different tRNAs that then will all interact with that single amino acid? Or do you have some cases where you have one tRNA that can interact with lots of different codons? And so it turns out that there's actually some of both. Sometimes you've got multiple tRNAs that will all have the same amino acid on them, but also very often you have one tRNA that can interact with multiple different codons. How can it do that? Well, the way it does that is what's called wobbling. Say, what the heck is wobbling? Wobbling is where you don't have a normal Watson-Crick base pair that forms. And this is particularly true at the first position. We're talking from 5' to 3' in the anticodon. So the third position that you have in the codon. And hopefully we all remember the genetic code, at least the overall structure. Very often that last nucleotide, the third nucleotide in the codon, seems to be pretty wishy-washy. It doesn't always have to be the same one. So it's this wobble hypothesis, which is what one of the things that Francis Crick came up with, is that the first nucleotide of the anticodon doesn't have to form a normal base pair with the third nucleotide of the codon. So for instance, here, our normal base pairing interaction would be UA, but with wobbling, you can actually have UG, and this is still a functional interaction. This is great, but it usually only works with a couple of possibilities, and it's going to be purine-pyrimidine base pairing. However, if you use these modified bases, inosine, for instance, it can interact with 3 different bases on the opposite strand. So that modification at that particular position in the anticodon allows that tRNA to interact with multiple different codons. And we now know through the high-resolution structures of the ribosomes, while they're elongating, that this is exactly right. The interactions there are not normal Watson-Crick interactions, whereas the first 2 positions in your codon always are normal Watson-Crick interactions. So what are some of these modified nucleotides? You've probably seen Ds or Us, etc. D is excuse me, dihydrouridine. All that is a modification to where you would normally have your double bond in the pyrimidine ring of uracil here. I don't expect you to remember all of these. Inosine is basically a deaminated guanosine. So instead of where you'd normally have an NH2 here, you just have an H. Some cases you actually have a thiouridine. We have a sulfur group instead of your oxygen. Dimethylguanine instead of hydrogen groups here. We've now got our CH3s. And my favorite down here, which is not in your textbook, is something called archaeocene. Archaeocene is, not surprisingly, only found in archaea and is probably important for the stability at very high temperatures that you find in some of these archaea, which is exactly what you would think of. A lot of these archaea were originally found as extremophiles. And I've been working again with Dr. Water-Royal to try and analyze exactly what this modification is doing to tRNAs. Turns out that a lot of the modifications that happen through this process, it's a multi-enzyme process. Again, many of those worked out by Dr. Water-Royal here in the chemistry department, making all of these various different modified ribonucleotides. And there are some genetic diseases where you're missing some of these modification enzymes, and in some cases also dietary supplements which you need to order to make some of these modified ribonucleosides. So it's a very important process, again, not terribly surprising since translation is so important. We mentioned inosine before. Inosine is probably the most important as far as we're concerned for this lecture, again, because it's very often at the first position in your anticodon loop, because that allows the base pairing to cytidine, uridine, or even adenosine. And so the weird thing is really this adenosine, because now I've got 2 purines that can actually base pair with each other. And normally you have to have purine-pyrimidine, again, purine-pyrimidine, but here in the case of inosine, you can have base pairing that forms here. You'll also notice that it's only 2 hydrogen bonds as opposed to the 3 hydrogen bonds that you would normally have between a GC base. Turns out that this is slightly different in bacteria and eukarya. In bacteria, inosine seems to be able to bind in more different positions, and you can have more different wobbles than you have in eukaryotes. What this means is bacteria can basically get away with fewer tRNAs than eukaryotes can. We've got about 40-some individual different tRNAs. Many bacteria only have about 30-some in terms of their different tRNAs. So that's the tRNA part. How does that tRNA, again, the 43 in humans and the 30-some in a lot of bacteria, how does it get the right amino acid attached to it? This is the job of the aminoacyl-tRNA synthetases. Here's an aminoacyl-tRNA synthetase here in gray, and I think this structure might actually have been solved by the Perona lab. I'm not entirely sure here, with the purple tRNA associated with it. And what you can see here, hopefully, is that our anticodon loop, which is down here, interacts with part of the aminoacyl-tRNA synthetase, and then it also interacts up here at the 3' end, which is where that amino acid is going to get attached to. So, not surprisingly, all of these aminoacyl-tRNA synthetases have to recognize different tRNAs. You could guess that if we've got 43 different tRNAs in humans, we'd have 43 different aminoacyl-tRNA synthetases. Turns out that's not the case. In most cases, you just have one aminoacyl-tRNA synthetase per amino acid. So what that means is each of those aminoacyl-tRNA synthetases can interact with multiple different tRNAs. And these are what are called iso-accepting tRNAs. So the aminoacyl-tRNA synthetase can interact with multiple different tRNAs. And what that means is that this anticodon loop interaction down here at the bottom is going to be not absolutely specific, whereas the interaction up here with the amino acid is going to be. So this is great. It's wonderful, what everybody thought until about 15 years ago. And then we were doing genome sequencing. And in genome sequencing, we found that there were a bunch of organisms that actually didn't have 20 aminoacyl-tRNA synthetases. They had fewer than 20 aminoacyl-tRNA synthetases, which was really bizarre. And it turns out that in a number of organisms, particularly in some of the archaea, they only have 17 aminoacyl-tRNA synthetases. And what happens is an aminoacyl-tRNA synthetase will put one amino acid onto that tRNA, and then that gets modified by a different enzyme later on. Why evolutionarily this should take place? Lots of theories. Ask Dr. Perona about why that should be. But this is what we found. And in fact, in the smallest archaeal genome found to date, it's, I think, down to 15 aminoacyl-tRNA synthetases, which is really kind of bizarre. It still uses 20 amino acids, but how it gets away with that is a really interesting question. So what are these aminoacyl-tRNA synthetases? They actually have 2 activities. The first activity is activating the amino acid. So we talked about activation at the very beginning of the class. We talked about coupled reactions, high energy intermediates, etc. So what's the high energy intermediate that's used for the aminoacyl-tRNA synthetases? It's taking the amino acid, and this is any particular amino acid here, and through hydrolysis of ATP, so it's getting this energy from ATP, hooking it up to ribose and adenine. This is that first step. This is the adenylation. This is why it's an aminoacyl-tRNA synthetase. First adenylates and then adds it to the OH at the 3' end of your tRNA. And this is now the high energy bond, which is originally made over here through ATP. Now it's hooked up through this ribose and adenine. That's again lost later on in the process. So ATP, you lose the phosphates here, but it gets attached to the amino acid, what's left, the AMP, and then AMP is lost when you charge, as they call it, the individual tRNA. So the aminoacyl-tRNA synthetase does both of these things. It takes the AMP from ATP, puts it onto an amino acid, and then puts that amino acid onto the tRNA. Now, This process, again, first we are going to modify our tRNA and put it onto your, modify the amino acid and then put it onto the tRNA. Now this charged tRNA is going to have to bind to your messenger RNA and then be used in translation. Once the aminoacyl-tRNA synthetase, again, here's one of those structures, aminoacyl-tRNA synthetase in blue, tRNA in green. Once this has been modified, this tRNA falls off, and now this is going to be interaction with the messenger RNA. Up here is the amino acid. And it turns out that the ribosome, and this has been shown quite clearly in a number of experiments, has no idea what's at the other end of the tRNA. All it does is figure out we've got our base pair interactions in the codon-anticodon loop, and whatever amino acid is up there, we're just going to attach it. So the real critical part of making sure that the right amino acid that is attached to the right tRNA is the aminoacyl-tRNA synthetase. So if you've got problems in your aminoacyl-tRNA synthetases, you're going to have all kinds of problems with translation, because the ribosome, only thing the ribosome checks is what's going on down here. It has no idea what's going on up here. So the specificity that you get for translation is really due to these aminoacyl-tRNA synthetases. So partly because of that, you want to make sure that these aminoacyl-tRNA synthetases are doing their job well. And all of you, I'm sure, remember structures of all your amino acids. No, I don't expect you to remember them for the class. But if you think about it, there are a number of amino acids which have extremely similar structures to each other. And this becomes a problem if your aminoacyl-tRNA synthetase is trying to figure out which one to attach that tRNA. And again, translation, fidelity is all going to be due, or mostly going to be due, to your aminoacyl-tRNA synthetases. So the aminoacyl-tRNA synthetases also have a proofreading activity, not unlike what we see with the DNA polymerases. Only here, the proofreading activity is basically chopping off any amino acid that's not the correct one. So we've got two different sites in your aminoacyl-tRNA synthetase, here, shown here as cartoons. Here's an amino acid which has just gotten added. And then, if it's the incorrect amino acid, you can go over here and get it chopped off. And now you've got the tRNA with a 3'OH that can be hopefully associated with this same aminoacyl-tRNA synthetase to put the right one in. And so, unlike the case where, again, in the proofreading that we see in DNA polymerases, where it's just looking at a structure of your base pair, here it's literally that specific amino acid. And that specific amino acid and the wrong amino acid is the one that gets chopped off. And this is particularly important for things like leucine and isoleucine. Just from the names, even if you don't remember the structure, you can think, they're pretty darn similar in terms of their structures. So this, what people will call mischarging, so remember the charged amino acid is the one that's got the amino acid on it, it will be the, for instance, isoleucine on a leucyl tRNA. That will then get cut off, and then leucine will get put onto it. That leucine doesn't get cut off. And so that's where our proofreading is coming from. Does that make sense? Yes? No? Confusing? OK, so let's look at a couple of those. I just mentioned isoleucine here, isoleucine and leucine, different positions of methyl groups. Valine is also extremely similar to isoleucine. Tyrosine and phenylalanine, in terms of their structures, are very similar to each other. And in the case of these aminoacyl-tRNA synthetases, the tyrosyl aminoacyl-tRNA synthetase will cut off phenylalanine. The phenylalanine tRNA synthetase will cut off tyrosine. And vice versa, the valyl tRNA synthetase will cut off isoleucine. The isoleucine tRNA synthetase will cut off valine if these guys are the incorrect amino acids which are put in. And again, it's specific to those individual amino acids. So mischarging I've mentioned already.
So with the editing activity,
yeah.
How does it know, does it do it by the structure of how it knows it's the incorrect amino acid, or does it do it by the different, I'm not quite sure.
OK, yeah, so basically how does editing work? Just to paraphrase your question. It is the structure. It's literally the structure, the chemical structure that we have here. So for instance, in the case of valine and isoleucine, it's the presence of this extra methyl group. And so it's literally, just like we've talked about for some of the enzymes, it's a very specific set of amino acids that is going to interact or not interact in the various different cases. So in the editing site, for instance, those will be the amino acids which will interact specifically with isoleucine and having this methyl group on it. And otherwise it wouldn't, because that's the appropriate amino acid for that particular aminoacyl-tRNA synthetase. Okay, more questions on editing and tRNA. Sorry, missed your hand there. Okay, so I mentioned this before. Again, the ribosome has no clue what amino acid is attached to your tRNA. As long as the tRNA is an appropriate one, codon-anti-codon interactions are correct, the ribosome will incorporate whatever amino acid happens to be there. And this was actually shown because you can purify RNAs. In this case, people purified the alanine-tRNA and then converted it to cysteine chemically, and the ribosome was perfectly happy to put in cysteines wherever alanines were supposed to be. Turns out this is an incredibly useful process, how people can, in fact, design new amino acids and change genetic code. Totally cool and interesting technology. Dr. Bartlett will be lecturing on that next term in the recombinant DNA techniques lecture. So just a quick aside there. So time for me to have a break. Time for you to answer some clicker questions. Post-transcriptionally modified nucleotide inosine is present in some tRNAs at which of the following positions? The 3' end of the codon, the 5' end of the codon, the 3' end of the anticodon, the 5' end of the anticodon, or I couldn't come up with anything else, so A and C. And again, vote early, vote often, and talk to your friends, or neighbors, or enemies, as the case may be. Still aiming for that 100%. Ten, 5, interestingly, it actually, excuse me, C and D were about half and half earlier on, and then D started to gain. So hopefully the discussions have helped with that. So that's not in the codon. Why is it not in the codon? Because, well, you don't find inosine in messenger RNAs, exactly. So the inosine is in the tRNA, so it's got to be in the anticodon. So 3' end or 5' end? 5' end, why? Because that's going to interact with the 3' nucleotide of your codon, because they're always anti-parallel relative to each other. And this is partly why I've been beating on the 5' and 3' and anti-parallel stuff. Yes?
Just on the modified amino acids, are they like the proteins or where they can go back and forth? What are they called? They're constitutional isomers of each other, tautomers.
Oh, tautomers of each other. So the question is are these tautomers? No, these are actually specifically chemically modified. So there are covalent bonds which have changed in order to get these. So there's no changing back. You're not going from inosine to guanine, et cetera. That'd be picking up amino groups, which is challenging to do. Yeah, once they're there, they're there, exactly. Okay, so now we have our charged tRNA, hopefully the right one because of the aminoacyl-tRNA synthetase. Now, finally, we get to make proteins. So how are proteins made? Proteins are made from N to C terminus, and quick reminder, talked about this thing in lecture 2. Amino group is at one end of any individual amino acid. The carboxy end over here is going to be the other end of the individual amino acid, but any protein as well is going to have an amino end and a carboxy end. How the protein is formed, you have the amino group of the next amino acid, which is going to attack the carboxy group here. You will remove this tRNA. Now this growing peptide is attached to this tRNA, and so on and so forth. Now, this is different from when we've talked about nucleotide polymerization, because nucleotide polymerization is just the nucleotide which is hanging out there. It's just attached to the nucleotides. Here, we're always attached to a tRNA in this elongating process. So here, where is that energy coming from for this? Originally, it came from ATP, but now it's in this high energy bond right here. Now, high energy means what? It means that when you have hydrolysis of it, you get a lot of energy. So this allows you to get polymerization to take place. So it's the hydrolysis of this high energy bond which allows formation of the peptide bond, which is what's between here, the C and the N, in order to have elongation take place. So this is translation. That's how translation works. Again, attacking the peptide, which is bound to one of the tRNAs, and then you hook it up to the next tRNA, etc. You're immediately going to ask, how do we start? How do we end? We'll probably get to that on Monday. But at least for now, let's talk about what does that. It's the ribosome. Similar to what we've talked about already for replication and transcription. Multiple steps. We have initiation, elongation, and termination. Probably most similar, at least conceptually, to what we see with transcription. So transcription, you initiate, you're going to start at a promoter, you're going to have elongation, and then termination. And you could also draw this as a loop here because we have a initiating ribosome, an elongating ribosome, a terminating ribosome, which then goes back to initiate and around and around and around. So we're mostly going to look at elongation today. We'll look at initiation and termination on Monday. Another way to look at this cycle is here, initiation, elongation, termination. Now, there are a couple of important aspects about this whole process. The main one is that it's the small ribosomal subunit which interacts with the RNA and gets all of the initiation together. And the main thing here is getting this initiator tRNA to the start codon, which is always what? AUG. Almost always AUG. Whenever I say always, you say, Dr. Stedman, there's got to be exceptions. This is biology. So the next thing that happens, once you have this initiator tRNA bound to the start codon with a small ribosomal subunit, then you have the large ribosomal subunit that associates here. It's that large ribosomal subunit which catalyzes the reaction that we just talked about, the attacking of the amino acids and elongating the chain. So here, once you have this associated, now this, I like to think of this as actually an amino acid polymerase, another way of talking about the ribosome, because it's going to polymerize amino acids. Then you have a new tRNA with the appropriate amino acid on it that comes in. You have transpeptidation that takes place. Again, that same reaction. The ribosome will move until it gets to a final termination, the stop codon. Then everything falls apart. Get a new messenger RNA, new small ribosomal subunit, new initiator tRNA, and the process continues. Mention large subunits. Oh, sorry, in the back. Okay, I'm sorry, I didn't quite understand your question. So it has to do with the moving of the messenger RNA in the ribosome? Yes. Oh, okay, yes. We'll talk about what we need that energy for in just a second. Yeah, actually probably maybe even on Monday at this rate. But yes, no, we'll talk about what you need that hydrolysis for later. But yeah, no, the actual energy for transpeptidation that is all then coming from those aminoacyl-tRNA synthetases, well noticed. But yeah, well, unfortunately, that's what happens when I've used things from different textbooks. They don't always match up completely. So that was supposed to be an overview, and again, well, yeah, but you're exactly right. Turns out that there's a huge amount of GTP hydrolysis that has to happen in translation, and that's why translation actually ends up being one of the most energetically expensive things to do inside the cell because of those extra bits and all of the ATP, which is being hydrolyzed by those aminoacyl-tRNA synthetases. So you've got some of both, which is actually happening there. Okay, other questions on this? Okay, so I mentioned the large and the small subunits of the ribosome. What do we mean by large and small subunits? And very often people will start talking about these S's, 16S, 18S, 70S, et cetera. What do we mean by all of these S's? And we'll talk about this again right at the very end of the class. They're called Svedbergs. What the heck is a Svedberg? So a Svedberg is a unit of measurement which has to do with sedimentation. And basically, the larger the S number, the bigger it is, because that's where it ends up in a particular centrifuge. And so when you do these centrifugations, 70S ends up being down here at the bottom for the prokaryotic ribosomes. And I say prokaryotic is because bacteria and archaea have these 70S ribosomes. It's made up of a 50S and a 30S subunit. Wait a minute, 50 plus 30 doesn't equal 70. It does for ribosomes. And that's because they're actually more compacted, and that has to do with where they're centrifuging in this centrifugation process. So if you get a ribosome, and particularly a ribosome that is not translating, you'll have small subunits and large subunits. These then have to come together, and those are going to be the active ribosomes. And you can do this, and this is what allowed Gobind-Khorana and Marshall and Nierenberg to do those experiments to figure out the genetic code in the first place, because you could purify ribosomes this way. In those ribosomes, we've also talked about the RNAs which make them up. The large subunit of a prokaryotic ribosome has 23S RNA, 5S RNA. The small subunit of the ribosome in bacterial and archaeal systems has a 16S RNA. Why do they call these S's? Because also from centrifugation. If you separate them from each other, they've got these different sizes. If you think about the tree that we looked at the very, very beginning of the class, the tree of life, the bush of life, almost all of that is based on these small subunit RNA sequences because all cells need translation and all cells use ribosomes. And these are all going to be similar sequences because they're going to have similar structures and similar functions. Eukaryotic ribosomes are a little bit bigger. So instead of the 70S, it's 80S, but also made up of two different subunits, each of them a little bit bigger, 40S and 60S. Again, 40 plus 60 doesn't add up to 80. But the small subunit ribosomal RNA is 18S. The large subunit is 28 with 5.8 and 5S behind my little clicker question up here. The 5.8, the 23, and the 18 are all made from that pre-ribosomal RNA that gets chopped up and then modified through all of the SNO RNAs. So they're made as one piece, but then chopped into different pieces, modified, and then put together to make the actual ribosome. And again, as I mentioned before, it's really the small subunit of the ribosome, both in prokaryotes and in eukaryotes, that interacts with the messenger RNA, gets the start codon in the right place, but it's the large subunit of the ribosome which is important for that transpeptidation reaction. What do these look like? And this has really been, well, I guess it's been about 15 years now, since the first high-resolution structures of the ribosomes were solved through, at that point, X-ray crystallography. If you look at the large subunit of the ribosome, and I think this is, I'm not sure if they're still using this particular image in the textbook, they talk about this as being a prokaryotic large subunit of the ribosome. It's actually an archaeal ribosome large subunit because archaea are so much cooler. But it's interesting, actually, because archaea and eukaryotes actually have a lot more similarities to them in translation than archaea and bacteria. So But be that as it may, turns out these structures are very similar to each other. The main thing to point out here is that it's almost all RNA. All of the gray in this structure is RNA, and all of the yellow is proteins. So all the proteins are sort of sitting around on the outside, holding the RNA in the right place. The active site, and we talk about the active site here, that's where the tRNA is going to be binding. You'll notice here there are no proteins anywhere to be seen around this. And a number of researchers, including Harry Noller, who I may get a chance to show you a link to in just a second here, had been studying the ribosome literally for decades, and they said it's all about the RNA. It's really the RNA. And it turns out you can get rid of all the protein in a ribosome, and it will still catalyze transpeptidation. It's pretty wimpy, but you still get that. And so it's really all about the RNA. And that's basically shown again here. Here is the RNA shown in lots of different colors here, mostly having to do with the conservation of each of these individual pieces. It's not just the 16S and 18S RNAs which are conserved. The large ribosomal subunits are also extremely well conserved throughout all of cellular life. And you can piece together all these individual bits and try and figure out how they're conserved relative to each other. And it turns out that the big difference between the eukaryotic and the prokaryotic ribosomes are just extra loops, extra pieces of RNA which are in them. It seemed to have very little to do with the actual activity here. And because these are also really well conserved, people will talk about the ribosome as being kind of a molecular fossil because they're so well conserved. That seems to happen. Translation is the same in every single cell that we know of, which the most parsimonious argument means that, yes, this was the original translation machinery going from the nucleic acid language to the protein language was a ribosome, and probably the very origins of life, at least the origins of cellular life, the origins of translation, you had a ribosome that looked a lot like this, and the ribosome's all RNA. So it means that a lot of the original cellular machinery, the invention of proteins, came after we had RNA. So again, it's all about the RNA is the take-home message here. If we now look at both the large subunits and the small subunit of the ribosome put together, and again, these are structures which have been determined literally in the last 15 to 20 years. Now we have both the large subunit down here, the small subunit down here, and where we have 3 binding sites for tRNAs. And this is shown here in the cartoon right here. If you look at the 2 subunits when they've come together, you'll notice you can barely see those tRNAs. So they're pretty well compacted on the inside, which makes sense when I was talking about the small subunit of the RNA interacting with the initiator tRNA. That initiator tRNA can't even get into the ribosome if both of the subunits are together. So you have to have first that association, and then the 2 subunits come together. The 3 sites are, again, incredibly creatively named: the E site, the P site, and the A site, the exit site, the peptidyl transfer site, and the aminoacyl site. So exit site, this is going to be where your tRNA is when it comes out of the ribosome. P is where transpeptidation takes place, and A is where you're going to have that next amino acid coming in bound to the tRNA. So what do these look like? Before that, one last question. How many different binding sites are there for tRNAs in the ribosome? We're trying to get 100%, right? We're going to get 100% on this? I certainly hope so. How many different binding sites? 1 through 5. And I know you're trying to mess with my statistics, right? 7, 5, sigh. Anyone who voted for 1 and 2 should probably come and talk to me, or you're, again, just trying to mess with me. So yes, there are 3 sites, the A site, the P site, and the E site. I'm not trying, wasn't supposed to be a trick question. So I know Stedman always asks trick questions. Maybe that's what people are thinking. OK, so I did want to just go through this animation. I think it's a really nice one. Again, it's the same group that did the animations of the DNA structures and replication and transcription. This is sort of that next step. So let's see if I can get this to come up. No, it doesn't want to go. Try over here. This one works better. Connect to it. Here we are. So this is that animation, again, from the DNA Learning Center. Here we have the, let me blow this up to full screen, the modified tRNA coming outside of the nucleus. And we have our ribosome chunking along with all of the individual aminoacyl-tRNAs that are associated with it. So all these little things floating around, the L-shaped molecules, are your tRNAs. Each of those, you'll notice, has a little red dot at the end. That represents the amino acid, which is going to get polymerized into the individual protein. Here's our small subunit, interacts with the 5' end, zooms along to the start codon. You have the large subunit, which is associated with it. Now, maybe a little hard to see. They should be different colors now, each of those individual codons going through there. Here are our tRNAs. Each, again, they're charged. They've got that appropriate amino acid, the little red dot at one end. We're missing, of course, the aminoacyl-tRNA synthetases in this particular process. The commentary here is actually pretty reasonable. Now, if you just pull away the small subunit of the ribosome, the large subunit looks like this. And again, it's chunking through 3 nucleotides at a time. If you now zoom into that crack, the only place which you can get a new tRNA to come in is at that A position. Again, if you think about where the small subunit of the ribosome should be, it's going to be sitting down right on top of where those are. And the only place you can get a new tRNA to come in and bind is going to be at that A position. Here, the individual amino acids are coming out through the ribosome and are eventually folding up into a structure. So there's that A site, the P site, and the E site, aminoacyl site, transpeptidation site, and the exit site. And this always has, it's a little hard to see here, but again, it's the tRNA that's attached to the protein. The tRNA, as you're building the protein, you're just moving that amino acid onto the tRNA, which is going to be at the P site. Each new one that comes in is going to be continuing to add to it. So it's only when you finally get to the end of your messenger RNA that you will have the protein actually being released. Neat secondary structure forming in our messenger RNA as we can look at it here in that whole process. So again, extension goes through a few more extra times than I probably would have. Now we have the protein. You'll notice that it's a peptide which is coming out of the ribosome, now slowly folding into an alpha helix. So as that's being made, it's really just stretched out amino acid. And then it's only once it folds into that 3-dimensional structure, it only happened on the outside of the ribosome. That was termination. Now our protein floats off and perfectly happy and ready to go. Let's stop this. Go back to our presentation. Oops, where are we here? Yep, great fun. So last 5 minutes, I want to talk a little bit about initiation and how initiation takes place. This is now initiation in bacteria. It's quite different than initiation in eukaryotic systems. So kind of mentioned this already. Really important part is getting the small subunit of the ribosome together with the messenger RNA and the initiator tRNA at the start codon. And I've been talking a lot about the initiator tRNA. The initiator tRNA is a separate RNA. So you're always going to have AUG, always, Most of the time you have AUG, vast majority of the time. AUG is going to be your codon, so you have an anticodon which is going to be encoding for methionine. It turns out that there are actually two different tRNAs that are going to code for methionine. One which is specific to initiation, and the other one which is specific to methionine that you're going to have in the rest of your protein, so when you have an AUG later on. So the initiator tRNA has to somehow associate with the AUG, and it has to be in the P site, because that's where you're going to be doing all of your extension. If you just, again, have the small subunit of the ribosome, then the P site is accessible. The large subunit comes down on top of it. You can't see that P site anymore. In bacteria, this methionine is actually a formal methionine, which marks that as being the N-terminal end of your protein. Not true in eukaryotic systems. And it's only once you have all three things together here, the messenger RNA, the initiator tRNA, and the small subunit of the ribosome, before you can have all of these associate with each other. So let's zoom into that small subunit of the ribosome and see how you get this association taking place. Again, this is specific for bacteria. In bacteria, you have what's called a ribosome binding site. A ribosome binding site is a specific sequence in your messenger RNA that's complementary to a sequence in the small subunit RNA in the small subunit of the ribosome. Also known as the Shine-Dalgarno sequence, because Shine and Dalgarno were the people who originally characterized it. And again, it's going to be base pairing with the small subunit of the RNA, and in this case, this is for E. coli, CU-CCU, which is going to base pair with AGGAG, and about 7 to 8 nucleotides, 3' to this ribosome binding site, you're going to have your AUG. So pretty straightforward way to have the association of your ribosome and ribosome binding sites here. We talked about polycistronic RNAs before. Polycistronic just means multiple different proteins are being made by one RNA. And the way that can happen is you have a ribosome binding site in front of each of those coding sequences. So the small subunit of the ribosome can bind here, get the initiator tRNA, associate, protein alpha in this case is made. There'll be a stop codon right here. So this will be an open reading frame here. There'll be another open reading frame, another open reading frame, making these 3 completely different proteins. So in order to do this, we also have more alphabet soup. So-called, sorry, yeah.
When bacteria are doing the polycistronic translation, are there multiple ribosomal units on there doing them at once, or do they disassociate and like that?
So the question here basically has to do when you're undergoing polycistronic translation, is it just one ribosome that'll hop on and hop on, hop on and hop off? We'll see, and this is also true for eukaryotic systems, usually any given RNA actually has a bunch of ribosomes that are associated with it. So, and the other thing which happens, so we'll talk about initiation factors next time, is translation is co-transcriptional in bacteria. So as the RNA polymerase is making this RNA, the ribosomes are going to bind to that immediately. And so as soon as one ribosome starts to translate this, another ribosome will come down and bind and start to translate from there as well. So I can't talk about initiation factors today. Have a good weekend. We'll see you on Monday.